The last lecture that we're going to have for this section is going to consist of DNA or genetic mutations. And there are some advantages to mutations, believe it or not. It's not completely bad because we get from these mutations, we derive evolution and genetic variation. There's a lot of disadvantages, though, because they can cause disease or even death. And proteins may not be made. So if that's an essential protein for something, then we would have a problem because of that. And the actual definition of a mutation is a permanent change in the DNA base sequence. In order for a mutation to be passed on, it has to actually happen in the gametes. Otherwise, there can be some external factors that cause mutations in other cells in the body. Okay, oops, I'm gonna put base here. All right, so the process that is being interfered with is, of course, protein synthesis, because that's our ultimate goal. Um, and then the cells that they affect, we've already mentioned somatic cells, which are all the cells in the body besides sex cells, and the gametes. Because if a mutation happens in the egg or sperm cell, then the mutation is going to be passed on to the offspring. What causes mutations? Well, they can be spontaneous. They can be caused by a carcinogen, which is a cancer-causing agent. We also call those mutagens. And these can include X-rays, smoke, UV light. We also get errors in replication. We'll put that up here. And errors during meiosis. So these are all things that we've talked about before, but now we're applying them. Okay, so what actually is happening? And when we talk about mutations, there's some specific types that we talk about. We talk about chromosomal mutations and we talk about actual gene mutations. And gene mutations include things called a substitution, and we'll look at each of these, which is when a base is replaced by another base in that DNA sequence. We could have a frame shift mutation, which includes insertions, actually inserting a base into the sequence, or deletions, which is the opposite, deletions. And that's when a base is going to be removed from the sequence. And because you have a base that's either added or removed, it's going to change the sequence that the DNA is read in. So it's going to change the actual codons in the RNA or in the DNA replication. Now, chromosomal mutations, we've talked about these before, and that includes non-disjunction. So when the chromosomes stay together. If we look closer at each of these, if we had some base sequences, and we'll start up here, and we're looking at this DNA strand unwinding. So this is our original strand up here, and the correct copy is put in. Well, in this copy down here, we're going to call the bottom our mutant copy because there is a base that is different. This would actually be called a substitution, because now if our code read CTGGAG, now it's being read as CTGGG. So that's going to change when it eventually gets coded into mRNA because it's going to change the bases that are read. Now, some substitutions are not going to cause a change because, as you saw on the genetic code sheet, there are many sequences that actually code for the same amino acid. So if there's a substitution that codes for the same amino acid, we're still going to get the proteins that are made. The problem is when it codes for a different amino acid. Now let's look at our frame shift mutations. And a frame shift is going to be just like we were reading a sentence, and now it's going to be off because we've changed something. So if the original sentence was the fat cat uh, sat, now because we had this deletion here, which is what this X is showing, now you can't even read it because now everything has moved over because remember the ribosome is going to read everything in threes. So it changes it and now it's going to read the three bases. Again, we can look at deletion down here, and if we see the CTGGAG, and in this case we have a deletion of two bases, now we're only left with this. Here we would have had a codon, and here would be a codon. Now we only have one codon, and it's still not the same as what was there. So we're not going to get the proteins that were made. And the other frame shift is an insertion. So now you have bases added into the code. So where we had CTGGAG, now we still have CTG. So if this were a codon and this were a codon, now we have CTG. That one's the same, but now we go CTG and GAG. We've actually added an entire codon, which is going to add a protein into the sequence, which is most likely not going to code for the protein that is needed. So there's your mutations, your gene mutations, which we're concentrating on in this unit, your substitutions and frame shift mutations.